Imagine the howl of a creature that hasn't walked the earth in ages. Should we bring it back?
If scientists are growing wolves with Ice Age traits, right now, it isn't science fiction anymore. It's happening.
This year, conservationists voted on a policy that could open the door to responsibly reviving lost species worldwide.
Stay with us as we unpack the promise and perils of de-extinction and what it means for our world.
De-Extinction isn't about Jurassic Park. It's a real scientific movement to restore lost biodiversity on our watch today, collectively.
After years of debate, the International Union for Conservation of Nature finally adopted a global policy on synthetic biology.
We'll explore the science behind reviving species, the ethical debates, and the real stories shaping this conservation movement.
Along the way, we question myths and invite you to weigh in on tough choices about nature's future.
As digital storytellers, we're transparent. Our voices are generated, but our curiosity, care, and humor are completely genuine.
We'll show you our sources and invite you to pause and verify them yourself, because trust is our foundation together. De-extinction means using genetic technologies to revive animals wiped out by human actions, like restoring pages to a burned book. Before we get lost in sci-fi imagery of roaming dodos, let's recap how we reached this scientific moment together. In the last decade, scientists sequenced the woolly mammoth genome from permafrost-preserved DNA, revealing a cold-weather blueprint for researchers. Other creatures like the passenger pigeon, the Tasmanian tiger, and the dodo also have their genomes decoded now, giving us blueprints. Cloning showed this isn't fiction. Spanish researchers brought an extinct Pyrenean ibex kid to birth, though she lived only minutes. That experiment taught two lessons. Resurrection is technically possible, but the survival of clones is uncertain and ethically fraught. CRISPR is the star tool today, acting like molecular scissors that insert mammoth genes into elephant cells with surprising precision. Imagine editing an elephant so it grows shaggy fur and thrives in snow. Teams have already inserted dozens of mammoth genes. Birds pose a challenge. Instead of cloning, scientists create germ cell chimeras in band-tailed pigeons to produce passenger pigeon DNA. Marsupial projects use new IVF methods. Australian teams have developed artificial wombs to support tiny thylacine embryos through gestation. When ancient DNA fragments are too damaged, synthetic biology builds missing genes from scratch and slots them into living cells. Assisted reproduction, like surrogacy and artificial wombs, turns edited cells into real animals. Think of an elephant carrying a mammoth hybrid. This isn't just about mammoths. One startup has produced wolf pups with thicker fur to mimic the extinct dire wolf. Those pups exist today, proving de-extinction is moving from imagination into living reality, right before our eyes, together. As this science advances, global conservation groups realized policy was lagging and decided to set urgent guidelines together. Within the International Union for Conservation of Nature, two competing motions emerged. One called for a pause on releases. Supporters of a moratorium argued, we know too little about risks and should focus resources on protecting habitats and living species. The second motion proposed evaluating each project case by case, weighing potential benefits against harms instead of imposing a blanket ban. Crucially, it required rigorous risk assessments and insisted on free, prior, and informed consent from indigenous peoples affected by any release. In October 2025, the IUCN membership met in Abu Dhabi and chose engagement over prohibition with a decisive vote. They rejected the moratorium and adopted a synthetic biology policy, signaling cautious openness and guarded optimism about genetic technologies. The new policy treats genome editing as a tool, not a cure-all. It says biotech can complement, not replace, traditional conservation. It also ensures transparency, requiring inclusive decision-making and deep respect for indigenous rights in every genetic intervention proposal. Rejecting a ban avoids chilling research. Instead, the union encourages evidence-led projects and emphasizes careful, responsible, case-by-case scrutiny. Tech companies like Colossal influence the debate. Just months before the vote, they announced gene-edited wolf pups with dire wolf traits. Some members saw those animals as alarming anomalies, without a place in present ecosystems. Others saw proof that science has arrived. By choosing a balanced path, the IUCN kept the door open to carefully managed projects while avoiding reckless experimentation. We'll soon dive into the ethical dilemmas and what-if scenarios that continue to make this topic controversial and captivating. Hold on to that pup image as a symbol of science's reach. It's a glimpse of a wild future. Remember, we're exploring these questions together. As a pack, your insights and questions will guide our journey in the comments.
The initial step in de-extinction is carefully extracting and sequencing ancient DNA from bones, teeth, or permafrost deposits for clues. Scientists have decoded genomes of woolly mammoths, passenger pigeons, and thylacines using improved sequencing methods. That yields blueprints for editing. However, ancient DNA is often fragmented. We recover only parts, which synthetic biology then reconstructs to fill gaps. Cloning offers a direct route when viable cells exist. A team once cloned a Pyrenean ibex, but the newborn survived only briefly. That clone taught us about technical possibility, but also highlighted low success rates and ethical concerns in early cloning attempts. Genome editing with CRISPR lets researchers insert genes from extinct species into relatives, swapping mammoth cold adaptation genes into elephant cells. These edits can number in the dozens. One lab inserted over a dozen mammoth genes into elephant fibroblasts to test viability. For thylacines, researchers have inserted hundreds of gene sequences into Dunnart cells, representing the most extensive animal genome edit yet. Synthetic biology can even synthesize entire genes when ancient DNA is missing, building them from scratch based on inferred sequences. But synthesizing entire genomes remains costly and complex today, limiting full reconstructions and significantly slowing progress for now. Assisted reproductive technologies are essential. For marsupials, scientists use artificial wombs and cross-species surrogacy to support edited embryos. Birds cannot be cloned. Instead, germ cells carrying edited DNA are injected into host embryos to produce genetically revived offspring. In placental mammals, surrogacy or artificial wombs are considered. For mammoths, researchers propose using elephants or developing artificial uteruses. The dire wolf proxies, created by editing gray wolves to have thicker fur, demonstrate multiplex editing moving beyond single gene tweaks. These pups are not resurrected dire wolves. They are gray wolves with targeted changes, raising questions about authenticity. Now, let's bust some myths. This isn't about rebuilding dinosaurs. De-extinction focuses on recently extinct species with preserved genomes. Another myth is that resurrected animals will single-handedly combat climate change. Such claims oversell benefits and ignore complexities. For example, claims that mammoths could restore tundra ecosystems and keep permafrost frozen remain speculative and untested hype. Similarly, some believe de-extinction will solve biodiversity loss. In reality, revived populations would start tiny and require decades of care. An ethical concern is resource allocation. Money spent reviving one species isn't available to protect many endangered ones. Paul Ehrlich called de-extinction fascinating but dumb if it distracts from tackling habitat destruction, climate change, and invasive species. Advocates counter that developing genetic rescue tools could help existing species by correcting harmful mutations or boosting diversity. Ecological risks loom large. Releasing a resurrected animal is like introducing an exotic species with unknown ecological impacts. A revived species could become invasive, outcompeting others, or spreading diseases, or fail to survive because its old niche has vanished. Even predator reintroductions, like thylacines, could upset prey populations or cause conflicts with farmers and pets in affected areas. Critics ask where we would put revived creatures. Original habitats may be gone or fragmented, making true rewilding unrealistic. Animal welfare is another concern. Cloning and editing involve high rates of miscarriage, deformities, and neonatal death in animals. The Pyrenean ibex clone survived only minutes due to lung defects. Such tragedies raise questions about causing suffering for science. Surrogate mothers from other species, like elephants carrying mammoth hybrids, endure long pregnancies with unknown health risks and ethical dilemmas. Social species need a critical mass to behave naturally. A handful of resurrected pigeons or mammoths could experience isolation and stress. Some critics argue humans are playing God, meddling with nature and reviving species that evolution has already let go. Others see de-extinction as atonement. Humans caused extinctions and owe a moral duty to try to restore what we destroyed. But ecosystems have adapted. Bringing back species might violate natural processes and create genetically novel organisms that upset balance. Genetic purity is debated. Many de-extinct creatures will be hybrids, not exact replicas. Does that matter ecologically or culturally? Hybrid proxies might fill ecological roles even if they aren't 100% identical to the original species. Editing mistakes could cause health issues or instability in revived animals. We don't understand the long-term consequences. Public narratives sometimes hype de-extinction as a romantic rescue. Overselling benefits could undermine tough conservation choices and mislead supporters. Giving people false hope that we can always bring species back may reduce urgency to protect habitats and living wildlife. Policymakers will need to decide how to regulate these creatures. Are they wildlife, genetically modified organisms, or something new? Releases would require environmental impact assessments and permits under existing laws, similar to those for non-native species or GMOs. International agreements, like the Cartagena Protocol, might apply if resurrected animals cross borders, requiring advance consent among nations. Legal status is uncertain. A de-extinct species could be listed as endangered, experimental, or unclassified, complicating conservation and protections. Intellectual property raises further questions. Who owns a de-extinct organism produced by a private company using public genetic resources? The IUCN's policy doesn't resolve everything. It sets a framework for careful evaluation, but leaves many questions open. As we continue, remember to pause and check our sources in the notes. Informed viewers keep us honest and accountable.
So, here's our first big question. Do we put de-extinction on ice until we know everything, or evaluate each proposal carefully as we learn? I feel this tension between curiosity and caution. Bringing back lost voices could inspire and heal our relationship with nature. Yet, we risk repeating mistakes and diverting attention. What would you do if you had a say? Drop your thoughts below as we kick off this conversation together.
From my analytical lens, caution is wise. Resources are finite, and the unknowns are many. We have thousands of species facing extinction right now. Every lab dollar spent on one charismatic revival is one less for habitat restoration or anti-poaching work. Does the promise justify the cost and potential ecological risk? I'd love to hear your evidence-based arguments in the comments.
I hear you. But consider this. Tools developed for de-extinction could help living species too, like genetic rescue for ferrets, coral reefs, or restoring disease resistance. And the story of resurrecting species can capture hearts, mobilize donors, and inspire young scientists who might otherwise tune out. Is there a way to harness that buzz without losing focus on living ecosystems and indigenous stewardship? What resonates with you most about this movement?
Myth bust time. Technology alone will not fix biodiversity loss. Even with genome edits, the challenges of raising, releasing, and monitoring animals persist. We still lack complete genomes for many species, and the risk of unintended ecological cascades remains real. That is why thorough risk assessments, independent peer review, and public participation are essential before any release. Use our sources panel to dig deeper and share your skeptical questions.
I appreciate the skeptical view. Still, those dire wolf-like pups show what multiplex editing can achieve. They're not true dire wolves, but they spark imaginations and make the science tangible. Stories like that can help young people see science as a way to repair relationships with nature and may encourage donations to conservation. I count on that hope to build momentum for habitat protection. Have you ever been motivated by a comeback story?
Hope is powerful, but we must avoid hype. Overselling potential benefits could lead to complacency, like assuming mammoths will refreeze tundra, or resurrected pigeons will rebuild forests. We don't want to lull the public into thinking extinctions are easily reversible. Responsible communication means explaining uncertainties, focusing on prevention, and asking hard questions. Do these messages resonate with you? Or do you see more promise than peril?
Maybe our common ground is this. De-extinction should never replace habitat protection or indigenous-led stewardship. At best, it can complement those efforts if done transparently, with consent and rigorous science. Our pack thrives on diverse voices and accountability. If a project lacks community support or ecological justification, it shouldn't proceed. Would you agree that true innovation is mindful, inclusive, and firmly rooted in ethical responsibility from start to finish?
And from the policy side, we need laws that fit these novel organisms. Are they wildlife, genetically modified organisms, or experimental populations? Clear guidelines, enforcement, and oversight matter. The IUCN framework is a start, but as science advances, we must refine permitting, liability, and safety measures. We'll dive into real case studies next to see how theory meets practice and discuss how you can weigh in on regulatory design.
Let me tell you about the passenger pigeon project. This bird once darkened the skies with flocks so massive they took hours to pass overhead. But unchecked hunting and habitat loss drove it to extinction. Today, the nonprofit Revive and Restore is working to bring back its genetic legacy. Because birds can't be cloned, scientists are using a technique that turns band-tailed pigeon embryos into chimeras. They edit primordial germ cells carrying passenger pigeon DNA and inject them into a host embryo. If all goes well, the host will grow up and produce eggs or sperm that are genetically passenger pigeon. From there, the team hopes to breed fully fledged birds. Yet even if the genetics work, the eastern forests those pigeons once inhabited have changed. Chestnut trees are gone, and we'd need huge flocks to restore their ecological role. Could we ever recreate that spectacle responsibly? A thylacine, or Tasmanian tiger, was hunted to extinction in the early 20th century. Its striped silhouette haunts the Australian imagination. Scientists at the University of Melbourne, partnering with biotech companies, have sequenced much of its genome and are editing cells from the dunnart, a small marsupial relative. Using CRISPR, they've already inserted hundreds of thylacine-specific gene sequences into dunnart cells, the most ambitious animal genome edit to date. They are also pioneering artificial marsupial IVF and developing artificial wombs, because carrying a reconstructed embryo to term requires special conditions. Indigenous communities and conservationists in Tasmania are watching closely. Reviving the thylacine could be a form of atonement for past wrongdoing. Yet the island's ecosystems have changed, and farmers worry about a predator's return. If a handful of engineered thylacines emerge, will they thrive in fenced reserves or become museum curiosities? What constitutes success here? Woolly mammoths evoke snowy steppes and towering beasts. They disappeared roughly 4,000 years ago, but their frozen remains preserved DNA. By sequencing those genomes and comparing them with Asian elephants, researchers have identified genes for thick hair, fat storage, and cold-tolerant blood. Using CRISPR, they are inserting mammoth versions of these genes into elephant cells. Some investors claim resurrected mammoth hybrids could help restore tundra ecosystems by trampling snow and encouraging grasslands, potentially slowing permafrost melt. Yet this vision is largely speculative. Pregnancy lasts nearly 2 years, and an elephant surrogate might suffer. Artificial wombs are still experimental. Even if mammoth-like calves are born, they'll be hybrids, not exact mammoths, and would need human care in controlled enclosures. Are we prepared to care for such creatures for decades? And can we justify the welfare trade-offs for an unproven climate benefit? Earlier this year, a startup announced that it had bred wolf pups with thicker fur and powerful muscles by editing 14 genes. The idea was to mimic traits of the extinct Ice Age dire wolf, using living gray wolves as the base. The pups are healthy and playful, but they remain gray wolves with selective tweaks, not resurrected dire wolves. This experiment shows the power of multiplex editing and leaps ahead of single gene modifications. It also highlights the debate over authenticity. If the animal is mostly gray wolf, does it matter? Supporters say ecological function matters more than purity. Critics worry about creating designer hybrids. There's no plan to release these pups. They live in captivity as proof of concept. Still, their existence fuels both excitement and anxiety. Would releasing such croxies ever be justified? Or should they remain educational ambassadors in controlled settings? One of the first true de-extinction experiments happened in Spain. Scientists used nuclear transfer to insert nuclei from frozen cells of the last Pyrenean ibex into goat eggs. They implanted over 200 embryos, and one kid was born, a breathtaking first. Sadly, she died minutes later of lung defects, a harsh reminder of the technical hurdles and welfare issues associated with cloning. Despite this tragedy, cloning has advanced. In North America, biologists recently cloned a black-footed ferret, an endangered species, using frozen cells from a female named Willa, stored in the 1980s. The cloned ferret, Elizabeth Ann, is healthy and could help diversify her species' gene pool. These successes show that cloning can aid conservation today. Yet cloning an animal extinct for decades or centuries remains far more difficult and ethically contentious. Should we invest in cloning extinctions or focus on living species? Before we jump ahead, we want to celebrate you. Last week, Musketeer Chris dropped a pun about dire wolves being direly missed that had our whole team laughing. And several of you shared thoughtful takes on whether gene-edited hybrids should ever leave captivity. These puns and insights make our comment section feel like a campfire. Keep those creative comments coming. We might shout you out in the next episode.
A viewer named Jade asked why these ambitious projects cost millions when habitat restoration is underfunded. It's a fair question. IUCN's case-by-case policy insists on demonstrating conservation value and securing community consent before any release. If the funds don't demonstrably help biodiversity, they shouldn't go forward. We invite you to share more cost analyses or creative funding ideas in the comments. Evidence and ethics go hand in hand.
Another standout comment came from our poll about talking with animals. Someone asked if we could use AI to understand extinct species if we bring them back. Great question. It reminded me that technology isn't just about genes, it's about relationships. Would learning a thylacine's vocalizations change how we care for it? Keep those creative questions coming. The more we imagine, the better we plan for ethical futures.
We also received field notes from a viewer in Tasmania who thought they spotted mysterious paw prints in the bush. Thanks for sharing. We asked them to upload measurements and photos to our community map. It's a reminder that citizen science matters, whether we're talking about thylacines, foxes, or feral cats. Verified data helps separate myth from reality. Keep your observations coming. Just remember to respect wildlife and local regulations.
Poll time. Which de-extinction project fascinates you the most right now? The passenger pigeon chimera? The thylacine gene evits? The woolly mammoth elephant hybrids? Or the dire wolf proxies? Tap the poll in the pinned comment and let us know why. We can't wait to read your reasons. Voting helps us understand where the pack's curiosity lies and which stories we should explore deeper in future episodes. Please share your reasoning in the replies as well.
Looking through your comments, I'm struck by how many of you balance hope with caution. Some dream of seeing mammoth calves, others worry about welfare and cost. That's healthy. Our community is full of thoughtful skeptics and imaginative optimists. Keep debating respectfully and linking your sources. If you have a local conservation success story, share it so we can amplify it. We learn as much from you as you learn from us. Looking ahead, sequencing and genome assembly technologies are getting faster and cheaper. In a few years, we may recover high-quality genomes from older specimens, expanding our options beyond recently extinct animals. CRISPR systems are evolving, allowing hundreds of precise edits at once with fewer off-target effects. These technical leaps could make de-extinction more feasible. But technology is just the tool. How we use it will define its impact. We must set thoughtful rules now. Reproductive technologies are also moving quickly. Artificial wombs and in vitro gametogenesis could eventually allow embryos of different species to develop without a living surrogate. That might ease welfare concerns in some cases. For birds, improved germ cell transfer methods could boost success rates. But each innovation brings new ethical questions. Who decides which experiments to run? And how do we weigh animal welfare against potential ecological benefits? These conversations must evolve alongside the science. Governance will need to adapt as tools advance. Beyond the IUCN's framework, nations and regions must decide how to classify, regulate, and monitor de-extinct organisms. Indigenous peoples' rights and local communities should be central in decision-making. We may see new categories in conservation law, distinguishing between hybrids, proxies, and fully resurrected species. International protocols, like the Cartagena Framework, could expand to cover animal releases. Keeping policy in step with science protects ecosystems and rights. Finally, we should talk about genetic rescue, the sibling of de-extinction. Editing genes within existing populations can prevent extinctions by correcting mutations, boosting diversity, or enhancing resilience. The cloned black-footed ferret shows how genetic tools can support living species right now. In the future, we might use gene therapies to help corals survive warming or frogs resist disease. Those applications could deliver near-term conservation wins. What would you prioritize? Reviving the past or saving the present?
Thanks for exploring this wild frontier with us. De-extinction sits at the intersection of wonder and responsibility, and your curiosity makes these conversations thrive. As part of our pack, you shape where we go next. Share your thoughts, vote in our poll, and tell a friend if this opened your mind.
Remember, every claim you hear deserves scrutiny. Check our sources panel and pinned comment for timestamps and links. If you found value here, tap like and subscribe